All right, so buckle up, everybody, because today we're taking a deep dive into the world of thalassemia.
Ooh, thalassemia.
Yeah, it's a blood disorder that affects millions of people all over the globe.
Wow.
And we're going to be focusing specifically on this really fascinating 2014 Indonesian research paper that delves into the nitty-gritty of mutations in something called the olobin gene, which is a pretty important player in how our bodies make hemoglobin, the stuff that carries oxygen all over.
It's like the delivery truck for oxygen in our blood.
Exactly. Yeah, so are you ready to get into it?
Absolutely. Let's dive in.
Okay, great. So this research comes to us from Gajah Mada University in Indonesia, and one of the things that really caught my eye was their focus on early detection of thalassemia carriers.
That's huge because early detection can open so many doors in terms of treatment options and family planning decisions.
Right, and especially in Indonesia, the paper mentions that the number of cases is increasing by like 8 to 10% each year.
Yeah, it's a growing concern, and that's why research like this is so critical.
For sure. So let's meet the researchers behind this paper,
Right.
Handiani and Ongo.
Okay.
They took blood samples from 10 individuals who were already identified as potential thalassemia carriers, and they honed in on this very specific part of the β-Globin gene called Exon 1.
So Exon 1 is like this instruction manual for building a protein.
An instruction manual. Okay,
Yeah.
I like that analogy.
Yeah, it's the blueprint that tells the cells exactly how to put together those building blocks that make up our bodies.
So kind of like the essential ingredients in a recipe.
Yes, exactly.
But wait, there's more.
Of course, there's always more.
So in addition to exons, we've got these other things called introns.
Right, introns are like those extra notes you scribble on your recipe card, maybe add a dash of this or a pinch of that.
Gotcha. So they're not like directly involved in making the protein, but they can still influence how the whole process goes down.
Exactly. They can tweak things here and there, and sometimes those tweaks can have unexpected consequences.
Oh, that sounds intriguing. So what did Handiani and Ongo find when they started digging into these exons and introns?
Well, using some pretty sophisticated DNA analysis tools, they found not one, but two mutations in exon 1.
2 mutations. Okay,
It-
I am officially on the edge of my seat. So spill the beans. What did they find?
Right, so the first mutation was a transition at nucleotide 59, where a T in the DNA sequence got swapped out for a C.
Okay, a T for a C.
Got it. And then the second mutation was a transversion at nucleotide 147, where a G was replaced with a C.
So we've got a T turning into a C and a G turning into a C. But hold on, what does all this even mean? Like, what are the implications here?
Well, the mutation at nucleotide 59, that's within the coding region, so it directly affects the amino acid sequence of the X-globin protein.
So that's like messing with the actual ingredients of the recipe.
Exactly, but then the mutation in nucleotide 147, that one's tucked away in an intron, so it's kind of lurking in the background.
Okay, so one mutation's a direct hit and the other one's more of a mystery. I see.
Now, here comes the really interesting part. Are you ready for this?
Hit me with it.
Despite these mutations, the final amino acid sequence of the β-Globin protein was completely normal, no changes whatsoever.
Wait, so we have mutations, but the protein comes out perfectly fine. How is that even possible?
It's like magic, but it's actually science. So the genetic code has some built-in redundancy, meaning multiple combinations of those three-letter nucleotide codes can specify the same amino acid.
So it's like using different words, but the sentence still means the same thing.
You got it. The DNA might look different, but the protein ends up being identical.
So are these mutations harmless then?
That's the million-dollar question, and that's what makes this research so fascinating. These mutations may be silent in terms of the protein sequence, but they might still be impacting the complex process of gene expression.
So those introns could be causing trouble even though the protein itself seems fine.
That's what the researchers are suggesting, and it really highlights the limitations of purely computational analysis.
What do you mean by that?
Well, you can only learn so much from staring at a computer screen. To truly understand the real-world consequences of these silent mutations, we need to take it a step further and conduct in vivo studies.
In vivo meaning?
In vivo means studying these mutations in living organisms. So this could involve animal models or even carefully designed clinical trials with human participants.
So it's like the difference between reading a cookbook and actually trying the recipe. Sometimes the results are surprising.
Exactly. Sometimes you need to see how things play out in a real living system to get the full picture.
That makes a lot of sense. So we're talking about moving beyond computer simulations and actually observing how these mutations behave in a living being.
Precisely. And that's where things get really exciting because we're moving from theory into the realm of real-world application.
I love it when science gets real. So we've covered a lot of ground already, from the basics of thalassemia to these mind-boggling silent mutations, but this is just the tip of the iceberg.
Oh, we're just getting started.
I can't wait to see where this deep dive takes us next, but for now, let's take a quick break, and then we'll be back to explore even more fascinating facets of this research and what it means for understanding and potentially treating thalassemia.
Sounds good. I'm ready for round two.
Me too. All right, we'll be back in a flash.
Yeah, it really is. And it makes you think about, what's the next step? Like, where do we go from here?
That is the question, isn't it? So if we were the researchers, trying to unravel this silent mutation mystery, where would we even begin?
Well, the researchers actually hinted at some potential avenues in their paper. They mentioned the limitations of in silico analysis, basically working with computer models.
Right, right.
So a logical next step would be to move towards in vitro studies.
In vitro. Okay, so we're talking about like test tube experiments now.
Exactly. They could take cells from individuals with these silent mutations, grow them in a lab, and then compare them to cells from people without the mutations.
Ah, so it's like creating this controlled mini environment to observe how the gene behaves in a living cell.
Precisely, it allows you to see if there are any differences in how the β-Globin gene is being expressed, or if that splicing process is being disrupted, the one we talked about earlier.
Right, right. So it's like zooming in on the action at a cellular level. That's pretty cool. But what other approaches could they take? I mean, we're talking about a pretty complex issue here.
Absolutely, and that's why it's so important to explore multiple angles. They could even go a step further and conduct those in vivo studies we mentioned earlier, where they look at the effects of these mutations in whole organisms.
Okay, so back to those in vivo studies, meaning?
In vivo, meaning we're looking at living, breathing creatures. This could involve using animal models or even carefully designed clinical trials with human participants.
Wow. We're talking next level investigation here, but I can totally see why it would be so important, especially given what's at stake. We're talking about people's health, their quality of life, even their future family planning decisions.
Absolutely, and that's why research like this, which focuses on early detection and understanding the root causes of thalassemia, is so crucial.
It's about empowering individuals with knowledge, giving them choices, and hopefully working towards prevention in the future.
Exactly. It's about taking this knowledge and translating it into real-world solutions.
And speaking of real-world solutions, let's zoom out a bit. Why is thalassemia such a big concern, particularly in places like Indonesia?
Well, for starters, it's not some rare, isolated condition. Thalassemia affects millions of people around the world, and as we mentioned earlier, Southeast Asia, including Indonesia, has a particularly high prevalence.
And it's not just about the numbers, right? There are significant social and economic consequences to consider as well.
You're absolutely right. Thalassemia can place a heavy burden on individuals' families and even entire healthcare systems. Treatment often involves regular blood transfusions and sometimes even bone marrow transplants, which can be very costly and demanding.
Yeah, it's a lot to cope with both physically and emotionally. And I imagine for developing countries where resources may already be stretched thin, the impact of thalassemia could be even more profound. Yeah.
Exactly. And this underscores why research like this, which emphasizes early detection and a deeper understanding of the genetic mechanisms involved, is so vital.
It's about giving people the information and the options they need to make informed decisions about their health and their future. And speaking of information, let's bring this back to our listener, the person on the other side of these microphones.
I agree. Let's make it personal.
Think about your own family history, your genetic heritage. Have you ever considered the stories that might be hidden within your own DNA? Have you ever thought about getting genetic testing for yourself or perhaps for future generations?
These are questions that are becoming increasingly relevant as genetic testing becomes more accessible and more affordable.
And it's not just about knowing your own risks. It's about understanding the complexities of genetics. The fact that even silent mutations might have an impact and the questions that come along with this knowledge.
It's a journey of discovery, and like all good journeys, it comes with its own challenges, surprises, and ultimately a deeper understanding of ourselves and the world around us.
Beautifully put. So, to our listener, we encourage you to keep exploring, keep asking questions, and keep marveling at the wonders of genetics. It's a field that's constantly evolving, and there's always something new to learn. And who knows, maybe someday you'll be the one making groundbreaking discoveries, deciphering the secrets of the human genome. But for now, let's take a moment to reflect on everything we've uncovered and then come back, ready to wrap up this incredible deep dive into thalassemia and the surprising power of silent mutations. Welcome back, everyone. I hope you had a chance to process all that information we've been throwing at you.
It's a lot to take in.
It is, isn't it? So we've explored, the intricate world of genes and mutations and even touched upon those silent mutations that can still have a ripple effect.
Right, like those sneaky ninjas hiding in the DNA.
Exactly. But before we wrap up our deep dive into thalassemia, there's one more layer I want to peel back, and that's the role of genetic testing.
Ooh, that's a timely topic, especially given our discussion of silent mutations and the questions they raise about, interpretation and potential consequences.
Exactly. Like, if a mutation doesn't even change the final protein, how do we even know if it's something to be concerned about?
Right, and what does that mean for things like carrier screening? It gets pretty complicated.
It does. So let's unpack this a bit.
All right, let's do it.
If someone undergoes genetic testing and is found to carry a silent mutation, like, how do you counsel them?
That's a tough one. Do you even tell them about it if it might not have any real-world consequences? What about the potential for anxiety and unnecessary worry?
Those are some really important ethical considerations, and I think they highlight the need for clear communication and careful consideration of the potential benefits and limitations of genetic testing.
Absolutely. It's not a one-size-fits-all kind of thing. You really have to approach each case individually.
For sure. So to our listener, if you're considering genetic testing, do your research consult with your healthcare provider and make sure you're comfortable with the potential implications of the results. Knowledge is power, but it's also a responsibility.
Well said, genetic testing can be a powerful tool, but it's essential to use it wisely and ethically.
Couldn't have said it better myself. And on that note, I think it's time to surface from our deep dive. We've covered so much ground today, from the basics of thalassemia to those fascinating silent mutations to the ever-evolving landscape of genetic testing.
It's been an incredible journey, and I really hope our listener has come away with a newfound appreciation for the complexity and the wonder of the human genome.
Me too. It's mind-blowing to think about all the intricate processes happening inside our bodies at any given moment and how tiny variations in our DNA can have such profound effects.
It really is, and it makes you realize how much we still have to learn.
So to our listeners, stay curious, keep asking questions, and never stop exploring the world around you, because there's always something new to discover. And until next time, keep those brain cells buzzing, and remember, every deep dive is a chance to expand your understanding and uncover new perspectives. Thanks for joining us.
It's been a pleasure.